Henry, thank you for being here today.
Thank you. My pleasure to be here.
Absolutely. How did you get into the field of GABA A variants?
Right. So I'm a biochemistry and molecular biology in training, and I've been studying GABAergic signaling for many years. And my PhD actually study how GABAergic signaling becomes inhibition. So what we call GABA is inhibitory is because of a chloride homeostasis. So chloride ion, how they distribute across the membrane of a neuron, is actually governing that signaling. So that's my background. And during my postdoc, I was studying how this GABAergic signaling is mature during brain development. And I know Dr. Rothenberg because of a collaboration when he has a line of work in his lab studying how after brain trauma, patients might develop epilepsy. And the way that how it develop epilepsy might be because of a loss of interneuron. So I was studying this interneuron, which are GABAergic, and that's a collaboration I started. One day, I was in his lab, in a lab meeting, and he was doing another line of work on rare disease patients. And there are some patients who has GABAergic homeostasis problem. The problem is that the patients have accumulation of GABA. GABA is a major inhibitory neurotransmitter, we know. But many of those patients, they have seizures. So this is a paradoxical where you have accumulation of an inhibitory signal, supposed to be sedated, supposed to be quiet and inactive. But in the contrary, these patients have epilepsy. So that got me, my basic science, nerdy side, very excited why you have this paradoxical changes, right? So we discuss. So I discussed with Dr. Rothenberg, and then one idea might be go back to my PhD study, understanding how that GABAergic signaling might be not working. Maybe GABAergic signaling was, compensated in some way, maybe impaired, and then let's take a look at those patients. So that's how we got started, and that's the. Then later on, we think about these patients have a gene mutation, particular gene mutation, monogenic disorder. So that's how the gene therapy study started. So that's how I started with
and so now your research is focused on gene therapy.
Yes. So our focus is developing gene therapy, what we call translational. So that will be translating our knowledge from bench to bedside.
And that process of gene therapy, if somebody's newly diagnosed, how would you explain it to them?
Well, the first thing we go back to the fundamental principle, like what get you that disorder, right? Because of the gene mutation. In Cure GABA A's situation, the patients have mutation, or what we call the variant, of their gene, of the GABA gene, right? So those are GABA receptor that are a composition of the GABA receptor, the different subunits, right? And they are the composition of the GABA receptor. One or multiple of those genes got mutated, right? And then the assembly of the receptor becomes a problem. And that the receptor is governing that inhibitory signaling. So the inhibition doesn't work in their brain. And then you have this EI balance issues, right? Excitation-Inhibitory balance is not balanced in that situation, right? So I would say that this is like from the beginning, from the genetic material when we inherited from our parents. Maybe or during that early development of the embryonic stage, mutation could happen, and that leads to this disorder.
Is there any way that patients can help you with your research?
Yes. So actually, what we are now doing is very, very close to the patient's efficacy group. For one reason is that because we want to learn from them, from the patients, what is their clinical manifestation? How do we use our tool in the lab? Meaning those mice, those cells, are those what we call the model can recapitulate the situation in patients. So learning from the patients how they, do they have seizures? Do they have developmental problem? Do they have behavior? Do they have sleep disorders? So those are the things that we want to understand. And then we have that in our mind, and then we go back to the lab and then develop models. that might recapitulate, right? We can mutate the gene the same way that the patients exhibit. And then we ask the question, do those mice or cells, exhibit some of those characteristics in patients? And if it does, then we got a good model, right? What's the use of the model? Then we can test some therapeutic. We can develop drugs, right? So in our case, we use gene therapy. So we develop gene therapy. We hope that gene therapy would work, and then we test it in those models. If those models change or getting better, then we predict that, okay, maybe when we put those same thing in the patients, then the same results hopefully will appear also.
Right.
Right.
And we're here at the 2025 Cure GABA A Conference, right?
Mm-hmm.
How is your work linked with Cure GABA A?
So we are already developing some potential therapy for Cure GABA A for some of the subunits. Yeah. So we know a lot of knowledge on GABA receptor and GABAergic signaling. And what we have not done is really to demonstrate that phenotype, that, like disease-relevant phenotype can be reversible. But we have done something in the other related disorders, still GABAergic disorders. They have epilepsy, so the patients also similar in terms of the clinical manifestation. We have done those other work in other indications, SLC6A1, SSADH deficiency. Those are the ones that we've been working for in a few years. And for those projects, we already got to a stage where we believe is quite close to clinic. So, and then through those experience, we know what needs to be done and what to expect. And also, we form Galibra Neuroscience is really the hope that we can translate those, bench to bedside very quickly. So once we've done that, then we just kind of like copy and paste using the same playbook and then do that. This is actually very common in the gene therapy field already. And some other programs, some other indications has been, like developing something similar, some formula that we want to do. There are nonprofit organizations, government organizations are actually doing something, helping us. Right? They make a playbook for us to follow. Right? What is the clinical trial design, or what will be the manufacturer for those therapeutics? So they want to make it a more like a plug and play, that kind of formula, if you will. So we are following that trend. We're following that development of that field, and of course, we are combining our knowledge on GABAergic signaling to make that happen.
That's amazing. So
Thank you.
Hopefully in 2026 we'll get to see you again at the Cure GABA A Conference, and we look forward to the progress that you guys have made.
Of course. Earlier this morning, we were talking to Monica, and we are already formulating some plan. We want to do that in 2026. So by the end of the year, next year, hopefully we'll see some progress in there.
That's amazing, Henry. I appreciate your time. Thank you so much.
Thank you.